Imagine a defense system where every single guard on duty is completely blind.
Oh, totally in the dark.
Right. They cannot see the enemy approach. They can't, read an ID badge. They can't scan a crowd.
Yeah, no visuals at all.
They have to identify lethal invading threats entirely in the dark, using absolutely nothing but physical touch.
I mean, it sounds chaotic, honestly.
It sounds like a terrible security system.
It does. But it is actually the most sophisticated security apparatus on the planet. And it's operating inside your bloodstream, like at this very second.
And that blind, touch-based defense is exactly what we are exploring today. Welcome to the deep dive.
Thanks for having me. I'm really excited for this one.
We've got an incredible stack of anatomy and physiology notes today, focused on the microscopic front line of our immune system, specifically antibodies and antigens.
The real, heavy hitters of the immune system.
Exactly. And if you have ever taken a rapid test or received a vaccine, or honestly even just wondered why a simple peanut can be life-threatening to some people, you have relied on this exact blind guessing game.
Yeah, the overarching story here is really one of recognition. We want to unpack the mechanisms of how our bodies actually know what belongs inside us and, well, what needs to be destroyed.
And I love the notes for this because sure, it sounds like a dry textbook topic at first glance.
And anatomy and physiology.
Yeah, but it's actually this wild story of contested definitions and really dramatic medical breakthroughs and intense scientific rivalries.
Oh, the history is fascinating. It's a field where our fundamental understanding of human survival has been completely rewritten several times over the last century.
So let's ground this in the textbook definition first, just to get our bearings. The classic analogy that everyone learns is a lock and a key.
The old reliable lock and key.
Right, so an antigen is the lock. It's essentially any substance that your immune system recognizes. And an antibody, which is this Y-shaped protein made by white blood cells, is the specific key that fits into that lock.
I mean, that is the modern sanitized version, yes. It's clean, it's easy to memorize for a test.
But the notes highlight that the terminology itself used to mean something entirely different, right? Like the word antigen is actually a contraction.
It is, yeah. It stands for antibody generator.
Which was coined around 1899 by this bacteriologist, Laszlo Detra. And back then, it literally meant a poison.
Right, a literal toxin.
Yeah. An antigen was defined as a toxic substance that forced the body to generate a reaction.
Which, that early definition makes total sense when you consider the terrifying diseases they were fighting at the time.
Like diphtheria and tetanus.
Exactly. They were looking for literal poisons. But that definition had to change because it turned out to be scientifically incomplete.
How so?
Well, in the early days, immunologists assumed a substance had to do two things simultaneously. It had to trigger an immune alarm, and it had to be bound by an antibody.
But those are two completely different jobs.
Very different jobs. As the field matured, researchers realized you actually have to split those properties apart. You have immunogenicity on one side and antigenicity on the other.
Okay, so immunogenicity is the ability to sound the alarm.
Yes, to provoke the immune system into action. And then antigenicity is just the physical ability to be recognized and bound by an antibody, regardless of whether any alarm goes off at all.
And the notes mention Carl Landsteiner actually proving this distinction in the 1920s using these things called haptens.
Landsteiner's work with haptens is just brilliant. So haptens are these tiny microscopic molecules.
Like incredibly small.
Right. They are so small that they basically float right past the immune system's radar. So they have zero immunogenicity on their own. They don't sound the alarm.
But if a scientist takes that happen and attaches it to a much larger carrier protein,
then everything changes.
It's like strapping a stealth bomber to a commercial airliner.
I love that analogy.
Right, because the stealth bomber alone doesn't trigger the radar. But once it's bolted to this massive, highly visible target, the military spots the airliner, sounds the alarm, and attacks the whole package.
And here is the really crucial part of that analogy. Once the immune system mounts an attack against that combined package, it learns the physical shape of the stealth bomber, too.
Oh, so it learns the hapten shape.
Exactly. It manufactures antibodies specifically designed to fit the hapten. And from that day forward, those new antibodies can find and bind to the naked, isolated hapten molecule all by itself.
So the hapten was always antigenic. It always had a physical shape that could be bound.
Yes.
It just needed help to become immunogenic and actually trigger that initial response.
Precisely. And this points to a massive paradigm shift in immunology.
True.
We had to stop defining these molecules by strict rules of what they are and start defining them by what they do.
Function over form.
Right, functional reform. In fact, if we hold too tightly to the classic Y-shaped definition of an antibody, we ignore entire branches of the animal kingdom.
Wait, what do you mean?
Take camelids, for example. So llamas, alpacas, camels.
Okay.
Classic human antibodies have a very specific structure. They have 2 heavy chains of proteins and 2 light chains.
That gives it the Y shape.
Right. But camelids produce what we call nanobodies. They're made of heavy chains only. There are no light chains at all.
So structurally, they completely violate the textbook definition of an antibody.
Completely. But functionally, they act as perfect keys.
They still bind to the antigens.
Yeah, they bind to antigens and neutralize threats just as effectively as ours do. So modern science defines antibodies by their neutralizing function rather than enforcing this rigid structural shape.
Okay, so if function is what matters, how did we first observe this neutralizing function in humans? Because the historical accounts in these notes are honestly incredibly dramatic.
Oh, the stakes could not have been higher. We really have to go back to Berlin in the late 19th century for this.
Right, to the diphtheria outbreaks.
Yeah, diphtheria was devastatingly fatal, especially for children. It was literally known as the strangling angel.
That is terrifying.
It was. And in Robert Koch's laboratory, a German physiologist named Emil von Behring and a Japanese bacteriologist named Kitasato Shibasaburo were just desperately trying to find a treatment.
And they realized that if they take an animal like, say, a guinea pig or a horse and inject it with a non-lethal dose of diphtheria toxin, something fundamental changes in that animal's blood.
Right, the blood serum of that surviving animal acquired this seemingly magical property.
Magical how?
Well, if they extracted that serum and injected it into a different animal that was currently dying of diphtheria, it would neutralize the toxin and cure them.
Ow.
They called this invisible protective substance antitoxin.
Which leads to this crazy moment on Christmas Eve, 1891.
Such a dramatic night.
They take this experimental diphtheria antitoxin serum derived from animals and they inject it into a dying child in a Berlin clinic.
And it saves the child's life.
It's literally the birth of serum therapy. I mean, they were using antibodies decades before anyone actually knew what an antibody physically looked like.
It was a monumental triumph for medicine, honestly. But it also became this major historical sore point regarding scientific credit.
Oh, right, because of the Nobel Prize.
Yeah. Behring went on to develop this into a massive commercial treatment, and in 1901, he received the very first Nobel Prize in physiology or medicine.
Alone.
Alone. He completely left Kitasato out of it.
Even though Kitasato was a co-author on all the foundational papers.
Right, and made arguably equal contributions to the discovery. It's a real shame.
But beyond the laboratory drama, this discovery forced a massive scientific question. Like, if serum has this magical ability to neutralize a specific toxin, how on earth does the body manufacture it?
And that question brings us to Paul Ehrlich. He actually coined the German word antikörper antibody around 1891.
Antikörper.
Yeah, and a few years later, in 1897, he proposed what he called the side chain theory. And I just cannot overstate Ehrlich's genius here.
Why is that?
Because in 1897, humanity knows absolutely nothing about DNA. We do not understand the atomic structure of proteins.
Right, it's basically the dark ages of molecular biology.
Exactly. Yet he somehow visualizes the lock and key mechanism. He imagines that immune cells are covered in a variety of these pre-existing chemical appendages, which he called side chains.
Okay.
And he theorized that an invading toxin flows through the blood, randomly bumps into a cell, and if its shape happens to perfectly fit one of those side chains, it triggers that specific cell to mass produce that exact side chain.
And then release it into the bloodstream.
Yes.
So those released side chains are what we now call antibodies.
Exactly. He basically mapped out modern cellular immunity entirely in his imagination.
That is wild.
He won a Nobel Prize in 1908 for that conceptual leap. But, you know, as brilliant as he was, Ehrlich's theory left this glaring mechanical mystery that would haunt immunologists for the next 60 years.
Right, the numbers problem.
The numbers problem.
Because if the body is relying on these pre-existing side chains, how does it know which specific keys to have ready for threats it has literally never encountered?
Right. Like a novel virus that just crosses over from an animal tomorrow. How could you possibly have a key ready for a lock that didn't exist yesterday?
And this sparked the great debate of 20th century immunology, right?
Right.
Instruction versus selection.
Yes.
So the instructive theory was championed by Linus Pauling, who was a heavyweight chemist in the 1940s, and frankly, Reading the notes, his idea makes intuitive sense to me.
It does. It really does.
Pauling argued that the body doesn't prefabricate billions of keys. Instead, it produces this generic blank slate protein. And when a new virus enters the body, this blank protein physically wraps around the invader, molding itself to the shape of the antigen.
The classic metaphor here is pouring hot wax over a signet ring.
Oh, that's a good word.
Right. The antigen serves as a template, physically instructing the antibody on how to fold and shape itself.
It's so elegant. It solves the numbers problem completely because you only need the genetic instructions for one generic wax-like protein, and it just adapts to whatever it touches.
It is a beautiful, highly intuitive, and completely incorrect theory.
Yeah, right.
Yeah, biology is rarely that accommodating. The instructive theory falls apart when you look closely at protein folding. So in 1955, a Danish immunologist named Niels Journey proposed the exact opposite, a natural selection theory.
Okay, so how does that work?
He suggested the immune system already contained a vast, chaotic diversity of antibodies before the antigen ever arrived.
And this gets formalized a few years later by Frank McFarlane Burnett, right, as the clonal selection theory.
Yes, the clonal selection theory. Burnett realized that every single one of your B cells displays one randomly generated antibody shape on its surface.
Just one.
Just one. And it just floats around in the dark, hoping to bump into something that fits. When a new virus enters your body, it doesn't teach your immune system anything.
It doesn't mold the wax.
No molding. It just drifts through the bloodstream until it blindly collides with the one specific B cell that, by sheer combinatorial chance, already happens to possess the perfect receptor.
That is crazy.
Right. And once that specific cell is found, it is selected. It activates, clones itself into an army of plasma cells, and just pumps out millions of copies of that one winning antibody.
Okay, I have to push back here because reading this from an evolutionary standpoint, that sounds incredibly wasteful and inefficient.
It sounds terrible, honestly.
You are telling me the body spends massive amounts of biological energy fabricating millions of random, completely useless keys just on the off chance that a specific lock might float by one day. Why wouldn't it just wait to see the threat and then build a custom defense?
Because building a custom defense takes way too long when the enemy's replicating exponentially.
Ah.
Bacteria and viruses mutate and multiply at terrifying speeds. If your immune system waited for an invasion to start designing a custom protein from scratch, you would be dead before the blueprint was even finished.
Oh, wow. Okay.
Evolution favored this massive, pre-existing library of random shapes because it guarantees that at least one cell will have a rough match ready to go the moment an invasion begins.
Okay, but then we hit the mathematical wall that the notes bring up because the human genome only has about, 20,000 genes in total?
Roughly, yeah.
And that's for everything. Building your brain, your heart, your bones. So how can we possibly have the genetic instructions to pre-cut billions of different antibody shapes? We simply do not have enough DNA to store all those blueprints.
And that was the ultimate paradox. But it was solved by Susumu Tonegawa in 1976. He discovered a genetic mechanism called VDJ recombination. And it is, without a doubt, one of the most remarkable parlor tricks in biology.
The notes describe this as genetic Lego blocks.
It's a perfect way to visualize it. So DNA is usually treated as this sacred, unalterable blueprint in our cells. You don't mess with it.
Right.
But not in B cells. Instead of carrying a massive dictionary of complete antibody genes, B cells carry a tiny, limited set of genetic fragments. These are labeled the V, D, and J segments.
Oh, it's literally like having a bin of Legos. You
Right.
don't have instructions for a million different spaceships. You just have like 50 red blocks, 50 blue blocks, and 50 yellow blocks.
Exactly. As a young B cell matures in your bone marrow, it does something totally extraordinary. It physically slices its own DNA.
It cuts its own DNA.
It cuts it. It takes one V segment, one D segment, and one J segment, throws the rest away, and splices those three together to create a brand new, totally unique genetic sequence.
That is mind-blowing. And
Yeah.
just by mixing and matching those limited fragments, you get incredible diversity.
But it goes even further than that.
Wait, really?
Yeah, once a B cell is activated by an antigen and begins rapidly cloning itself, it undergoes a process called somatic hypermutation.
Somatic hypermutation.
Yeah.
Does it intentionally mutate itself?
It does. The cloning process is deliberately sloppy. As the B cell divides, it introduces tiny random typos mutations into the specific DNA region that codes for the antibody's grasping arms.
But why would it risk breaking a key that already fits the lock? If it found the virus, why mess with the key?
Because it's looking for an even tighter grip. Most of those typos will make the antibody worse, sure, and those clone cells will just die off. But a few typos will slightly alter the shape of the receptor so that it binds to the antigen even more aggressively.
Oh, I see.
Those superior clones receive survival signals and completely outcompete the others. It is literal microevolution happening inside your lymph nodes in real time, just fine-tuning the fit until it is absolutely perfect.
That is staggering. The body splices its own DNA and then intentionally mutates itself to build a better weapon.
All to solve that numbers problem.
So after all of this splicing and mutating, the body finally has this massive army of plasma cells pumping out millions of perfect antibodies. They flood the bloodstream.
Right.
What happens next? Because there's a very pervasive myth about what antibodies actually do to the invader once they find it.
Oh, the assassin myth, yes. Most people picture antibodies as these microscopic soldiers hunting down viruses and, stabbing them to death.
But the antibody itself doesn't actually destroy anything, does it?
Very rarely. Antibodies are mostly just tags. They're signal flares for the heavy artillery. A primary mechanism for this is something called opsonization.
Opsonization.
Yeah. Let's say a dangerous bacterium enters a wound. A specific class of antibody, called IgG, will bind to the antigens on the surface of that bacterium, completely coating it.
But the antibody isn't hurting the bacteria, it's literally just stuck to it.
Right, but that coating makes the bacterium highly visible and incredibly appetizing to phagocytic cells.
Which are like the immune system's garbage disposals, like macrophages.
Exactly. A macrophage might ignore a naked bacterium, just float right past it.
Okay.
But when it sees one coated in IgG antibodies, it aggressively engulfs and digests it.
It's like pouring hot sauce on a bad guy.
Hot sauce.
Yeah, the hot sauce doesn't kill the bad guy, it just makes the monster who eats bad guys want to eat him faster.
There's a very vivid way to put it, but yes, exactly. Antibodies can also trigger the complement system.
What's that?
This is a cascade of proteins floating dormant in your blood. When an antibody binds to a bacterium, it acts as a landing pad for these complement proteins.
Oh, so we swarm it.
They assemble on the antibody's tail, they drill into the bacterial membrane, and they physically punch a hole in the cell, causing it to explode.
Wow. So the antibody is basically just the laser designator calling in an airstrike.
Perfect analogy.
And this tagging and clumping behavior is actually the entire basis for blood typing, isn't it?
Yes. Carl Landsteiner won a Nobel Prize in 1930 for discovering the ABO blood group system, which is honestly the most literal life-or-death demonstration of this blind recognition system we were talking about.
So if you have type A blood, your red blood cells naturally display the A antigen on their surface.
Right.
But your blood plasma is heavily patrolled by anti-B antibodies. So what happens if there's a mix-up at the hospital and you receive a transfusion of type B blood?
Well, your anti-B antibodies blindly bump into those new type B red cells. They recognize the foreign antigen instantly and they latch on.
Because they're the lock and the key.
Exactly. And because antibodies have two arms, they can grab a foreign cell in each hand. Then another antibody grabs one of those cells and links it to another.
They start gluing the foreign red blood cells together.
Right. It creates a massive sticky web of trapped cells. This is called agglutination. Inside a test tube, you can actually see the blood physically clumping together into sludge.
But inside a human vein...
Inside a human vein, that clumping causes massive blockages, kidney failure, and death.
Yes.
The immune system is doing its job perfectly, though. It is just reacting to a well-intentioned medical procedure as if it were a massive parasitic infection.
Man, there is another really fascinating example of antibodies moving between people in the notes, and it involves pregnancy. You mentioned earlier that IgG antibodies coat bacteria, right?
Yes.
But they also have a unique superpower regarding the placenta.
They do. IgG is the only class of antibody small enough to cross the placental barrier from a mother to her fetus.
Which basically provides the baby with a temporary borrowed immune system.
Exactly. This is called passive immunity. The newborn hasn't encountered any real-world threats yet, so its own B cells haven't done any of that genetic splicing and mutating we talked about.
Right. Its library is empty.
By crossing the placenta, the mother provides a temporary shield of pre-made antibodies to protect the infant for its first few months of life.
Just to contrast that, active immunity is what happens when you get a vaccine or actually catch a virus yourself.
Right. Your body does the heavy lifting.
It makes its own antibodies and critically creates memory cells so you can produce more later. The passive immunity from the mother eventually wears off because the baby doesn't have the blueprints to replace them.
That's the mechanism. But, this placental crossing has a really tragic dark side, too. It's the driving force behind Rh disease, or hemolytic disease of the newborn.
So this happens when a mother has Rh-negative blood, meaning she doesn't have the Rh antigen, but her fetus inherits Rh-positive blood from the father.
Yes. And during the delivery of that first baby, the mother is often exposed to a small amount of the baby's blood.
Right.
Her immune system, doing exactly what it evolved to do, sees those Rh-positive cells, recognizes the Rh antigen as foreign, and spends the next few weeks manufacturing anti-Rh antibodies.
But the first baby is already born and perfectly fine.
They are, but the mother's immune system is now heavily armed against the Rh antigen.
Oh, no. So she becomes pregnant with a second Rh-positive baby.
Those small IgG anti-Rh antibodies will cross the placenta, just like they're supposed to. But this time, they recognize the fetus's own red blood cells as foreign invaders.
That's awful.
They bind to them, tag them for destruction, and it can cause severe anemia or heart failure in the fetus.
It is friendly fire on the most devastating level. The system doesn't know it's attacking a baby, it just feels a lock that matches its key.
Exactly.
Which raises a huge question for me. If this system is entirely blind and relies on random mutations, how often does it accidentally build a key that fits our own organs?
Frequently enough that it has an entire medical classification. Autoimmune disease.
Wow.
This is the inherent danger of having a system that randomly generates billions of keys. Sometimes, the body's own tissues are completely mistaken for the enemy.
The notes highlight a mechanism called molecular mimicry, specifically looking at rheumatic fever.
Molecular mimicry is a fascinating and honestly really unlucky phenomenon. Let's say you catch strep throat, a group A streptococcus infection.
Yeah.
Your immune system generates a fantastic antibody that perfectly neutralizes the strep bacteria. You recover.
Great. But the physical shape of the strep antigen happens to look remarkably similar to the proteins that make up human heart valves.
Right. And remember, the antibody has no eyes, no brain, and no context. It is just a sticky protein floating in the dark.
So it bumps into your heart valve.
It feels a molecular shape that closely mimics the strep bacteria it was trained to hunt. And it attacks. It triggers the complement bomb squad and the macrophages to destroy your own heart tissue.
So the system didn't malfunction per se. It was just tricked by a molecular doppelganger.
Exactly. It's doing its job, just on the wrong target.
And the notes also detail cases where antibodies don't just destroy our cells, they accidentally turn them on. Like in Graves' disease, where an antibody perfectly mimics the shape of a thyroid-stimulating hormone, causing the body to radically overproduce hormones.
All of this really chips away at the common misconception that more antibodies are automatically a good thing.
Right.
And perhaps the most sobering proof of that is a phenomenon known as antibody-dependent enhancement, or ADE.
BDE. This sounds like the immune system actively switching sides. How does an antibody enhance an infection?
It is deeply counterintuitive, and the most notorious case study involves the dengue virus. So dengue exists in 4 distinct strains.
Okay.
If a mosquito bites you and transmits strain number 1, your body mounts a defense, splices its genes, and produces a highly specific antibody that perfectly locks onto and neutralizes strain 1. You recover, and you have lifelong immunity to that specific strain.
But what happens if, years later you get infected with dengue strain number 2?
Well, your immune system sees the new virus, realizes it looks an awful lot like strain 1, and deploys all those old antibodies. They flood the bloodstream and bind to strain number 2.
Okay, so it tags them.
It tags them, but because the new strain is just slightly different, the old keys don't fit perfectly. They grip the virus, but they fail to neutralize it.
So they are stuck to a live, active virus.
Exactly. Now, recall your hot sauce analogy from earlier.
The macrophage comes to eat it.
Right. The antibody is a signal flare. A macrophage arrives, sees the antibody tag, grabs it, and pulls the whole package inside itself to digest it.
But the virus isn't dead?
The virus isn't dead. And worse, dengue is a virus that specifically wants to replicate inside macrophages.
Oh my God.
The antibody essentially acts as a Trojan horse. It hands the live virus the exact key it needs to comfortably enter the very immune cell that was supposed to destroy it. The virus replicates massively, causing a much more severe, often fatal form of the disease.
That is terrifying. And this isn't just theoretical biology, right? This actually caused a massive public health tragedy involving a vaccine.
Yes, it did. Between 2017 and 2019, the Philippines rolled out a dengue vaccine called Dengvaxia. Now, a vaccine's job is to simulate a first infection.
So for children who had previously caught dengue in the wild, the vaccine works safely.
Yes. But for children who had never been exposed to dengue, The vaccine acted as their first exposure, priming their immune systems with those initial antibodies.
So when those specific children later encountered real wild dengue mosquitoes?
ADE occurred. Because of the vaccine's antibodies, the real virus was ushered directly into their immune cells. The vaccine actively increased their risk of severe, life-threatening dengue.
Oh, man.
The entire rollout had to be suspended. It is a really harsh reminder that in immunology, context is everything.
Yeah.
The wrong antibody at the wrong time is literally a weapon handed to the enemy.
That is incredibly heavy. But I want to pivot because the flip side of this is that once we understood exactly how this lock and key system works, we learned how to hijack it for modern medicine.
We did.
We have to talk about monoclonal antibodies.
This is perhaps the greatest practical application of immunology in history. So in 1975, Georges Kohler and Cesar Milstein wanted to manufacture specific, pure antibodies in a lab, but they faced a biological wall. B cells, the cells that make antibodies, die very quickly when you take them out of the body. You can't just grow them in a petri dish.
So how do you build an antibody factory if the workers keep dying?
You perform a piece of biological alchemy called the hybridoma technique.
Hybridoma.
Yeah. They took a mortal B cell, one that had been trained to pump out one specific perfect antibody, and they forcefully fused it together with a myeloma cell.
And a myeloma cell is a cancer cell, right?
Specifically, a cancer of the plasma cells.
See.
Cancer is characterized by runaway immortal cell division.
Okay, I see where this is going.
By fusing the two together, they created a chimera, a hybridoma. This new cell inherited the infinite lifespan and rapid replication of the cancer cell, combined with the exact genetic blueprint from the B cell to pump out one specific, perfectly identical antibody forever.
They literally cured the B cell of its mortality using cancer.
And for that, they won the 1984 Nobel Prize. Because once you can grow vats of immortal cells producing a pure targeted antibody, you completely change medicine forever.
It is everywhere now. I mean, if you know someone taking Herceptin to target breast cancer cells or Humira to block the inflammation in rheumatoid arthritis.
Or even the highly publicized monoclonal antibody cocktails given to severe COVID-19 patients.
Right. Every single one of those therapies descends directly from this technique. We are manufacturing vats of custom-cut keys to turn off specific locks in the human body.
We basically took Burnett's clonal selection theory and weaponized it to save lives.
It really is an unbelievable journey. I mean, we started in the 1890s where an antigen was just some mysterious poison, and scientists were injecting horse serum into dying children on Christmas Eve.
An act of sheer desperation.
Totally. And we arrived at an understanding of VDJ recombination, the knowledge that our bodies literally slice and mutate their own DNA to build an infinite armory against the unknown.
A defense system navigating purely by touch, shaping the entire course of human survival.
It's wild. Next time you take a rapid test or deal with a seasonal allergy, just remember that it's all governed by these blind guards feeling for intruders in the dark.
You know, before we wrap up, I just want to linger on that hybridoma technique for a moment.
Yeah, go for it.
We discussed how monoclonal antibodies, some of our most precise life-saving medicines, are manufactured by fusing a highly specific immune cell with an immortal cancer cell.
Right.
It is a profound biological paradox. Think about it. To create our greatest pharmaceutical shields, we literally had to harness the runaway destructive immortality of cancer, which is arguably our body's deadliest internal enemy.
That is intense.
It really makes you wonder, what other destructive, chaotic forces in nature are just waiting for us to understand them well enough to repurpose them as our greatest cures?
That is definitely something to chew on. Well, thank you for joining on this deep dive into the microscopic frontline, and thank you, the listener, for coming along on the journey.